Hello, I'm in the lab to do the Enzyme Lab now. So you want to, yeah, you can see, it's right here. So you want to make sure you have your lab manual. I'm actually going to be starting with the supplies we have to work with, and I'll be explaining some of the equipment too. So you're going to want to go to page 50 of your lab manual, which explains, actually has a list of materials. We'll go through all that, and then it's going to explain how the computer gets used that we have. So I'm going to tilt the camera down so that you can see some of these materials that I have. Okay, so what we've got here, this is our yeast, which I'm going to mix with some glucose, and I'll do it right now. Remember, the yeast is what we're calling the quote-unquote enzyme for our experiment. We're going to wake the yeast up by putting them or mixing them with this glucose solution. Just like if you do baking, you kind of have to wake your yeast up. Give them some sugar to eat. So I'm going to pour the yeast in. I'm going to stir them around. Give them a few minutes, and you'll start to see eventually they will start getting very bubbly. And for the rest of the lab, this stuff is the enzyme. So this is your enzyme. We've also got the hydrogen peroxide, which is in this container here. I know it just says repipette 2, but up here there's a label that says 3% H2O2. So this is your substrate in this bottle. We've got test tubes that will label and set up. Each test tube will be for like a different test of the enzyme plus the hydrogen peroxide under different conditions. And over here, isn't that looking real smooth? This is the oxygen sensor. I like to call this the magic wand. This is what we're going to use to measure the products of the chemical reaction. In other words, we measure how much oxygen is made in the reaction that we're doing, looking at how much hydrogen peroxide the enzyme can break down. The oxygen sensor is hooked up by this cord to this little computer over here. And you'll see that if I turn the computer on, you can barely see anything, right? I can't really see it, so I'll have to take a picture, but basically there's a table over here to enter values into, and when the oxygen sensor is actually turned on, it will record data the whole time, and then there's a graph over here that we can plot what's going on. So the next thing I'm going to do is show you, I'm actually going to work through the steps starting at the bottom of page 50. I'm going to go back here. So I'm going to start over here at page 50, and I will go through page 51. The first set of reactions that we do, the first experiment really is looking at how does the amount of enzyme that you apply to your reaction affect the amount of oxygen produced. So, and what is the effect of enzyme concentration on your reaction rate? That's the easy way to say it.